Luyi Tian is from Walter and Elisa Hall Institute of Medical Research of University of Melbourne in Australia. Yeah, so let's welcome Luyi to give their story on COVID-19, the asymptomatic COVID-19 patients. Yeah.
Okay. Thank you, Xue. Thank you. Also want to thank the organizers for letting me to give me the opportunity to speak in this wonderful conference. So today I want to give you a very brief overview due to the short time. Well, a very brief overview of this COVID-19 project that I'm collaborating with my Chinese collaborators. So I hope you, due to this, because of this is a single cell conference, I hope you can forgive me for not giving a background introduction to directly jumping to the result. And I hope everyone can agree that single cell sequencing can be a very important tool to understand the COVID-19 disease. And but still, I want to give a very brief introduction to why we're doing this. What we want to understand is what happens between the different conditions and why some people don't have any symptoms after they get infected, and why some others becomes a severe patient and goes to the ICU. So and what we did is we have a cohort of a bit small cohort of three healthy donors, and one severe, 7 moderate patient, and 2 asymptomatic patient. But for each patient, we have multiple samples from each patient taken at multiple different time points, and so this gave us 34 samples in total, and we take PBMCs and perform the single cell RNA sequencing together with the PCR sequencing to look at the repertoire profile as well. So this is a general overview of the data analysis. After QC and removing the dead cell, we can still get quite a lot of cells from all the patients. So this is the UMAP of all the cell type of the integrate data of all the patients from all the time point. I won't go to detail into each cell type, but you can say that on the right hand side is the dot plot of the markers, of some of the markers that we used to distinguish all the major cell type. I want to go into much detail about this plot because today we want to be a bit more focused on the T, NK, or lymphocyte cell subsets because we find some interesting result here. And what we did is to extract all the T, NK cells from the total population and redo the data integration and the clustering to perform a more detailed data analysis. And by doing so, we can find some rare cell types that we couldn't be found in the previous analysis that contains all the cell type. For example, we find some rare immature NK cell that in this population. And the first thing we looked at when we after we do this analysis is that we look at the proportions or the distributions or the abundance of the different cell types. And here are some examples that we found quite interesting, that in the asymptomatic patient, they have some higher abundance of some NK cells. For example, these few NK cell subsets, and we also sorry, I forgot to say here. We also find a lymphocyte that we couldn't really distinguish from the known populations, and also we find on the contrary, in the severe patients, there are some other NK cell types that enriched. And interestingly, from another independent project with a larger cohort, we also find this NK cell type to be increased in patients even after 6 months post infections, which is quite interesting. And then next we look at this new, I can't really say it's new, but it's a real cell type that we couldn't be annotated with a known with the known the databases. If we use single R, a single R classifies the cells as Th2 cells, and indeed it expressed some Th2 cell markers, but it lacked the CD4, CD8, which are very important T cell markers, and it also expressed a few NK cell markers as well. And most interestingly, expressed a gene called TNFRSF19. This is not a gene that typically expressed a lot in immune cell, and on the contrary, actually, this gene is expressed in epithelial cells. Also, thanks to all the people who contribute to the human cell lung atlas, which is a very great tool that we found, and we curated these genes in this human lung cell atlas, and not surprisingly, we didn't really find any expression of these genes in the immune cell subset, which is the light green part. But we find that this gene is expressed in some epithelial cells, for example, platelet cells and club cells, and also these cells also express ACE2, which is we all know is the entry of the virus. And we don't want to make any unsolicited assumptions, but we just want to point out this finding is quite interesting. And we also performed a differential expression analysis between different conditions, and interestingly, we found some upregulated genes that also been found in other studies. Although we only have one severe patient. For example, we found a massive increase in the type 1 interferon-related genes in severe patient, and actually we also find these genes upregulated as well in early moderate patient, and it decreases with time, but not as high as in the severe patient. So it seems the severe patient is really in a hyperinflammatory state that express a massive amount of these genes. And apart from that, interestingly, we also find that in multiple different cell types, in the asymptomatic patients, there's upregulation in interferon gamma, which is also very important genes. And apart from that, we also find in severe patients there are some other chemokines, cytokines that are upregulated in symptomatic patient. For example, the XCL2 and CXCR4, which are all chemokines and cytokines that are upregulated in the asymptomatic patient, especially in the NK subsets, which is also quite interesting. And apart from that, because we perform the TCR PCR sequencing, we can also look at its repertoire, its clonal, its PCR clones. And the first analysis that we did is simply look at the clonal abundance and the clonal expansion, and we look at the different samples from different conditions, and we found that there's a massive TCR clonal expansion in the moderate patient, especially in the early time point, which makes sense because as the time goes on, the patients start to recover from the disease, and the clonal expansion kind of decrease, which makes sense. And however, we don't find a significant clonal expansion in asymptomatic patient and in severe patient, especially when you compare it to the moderate patient. You can see that from the pie chart here, where the color indicates the level of clonal expansions. And apart from that, we also look at the clonal expansion TCR profile in terms of cell types. On the right-hand side is the EU map that colored not by cell type, but colored by the clone type, and the way we select the top enriched clones for each conditions. And of course, we say that in the moderate patients, there's a lot more clonal expansion. You see larger clonal node size in moderate patient. However, in the asymptomatic patients, although there's not a high clonal expansion, we find that the clonal expansion seems to preferentially happens in the effector CD4 T cell instead of CD8 T cell, which is also a very interesting finding. And to conclude, what we find that is the asymptomatic condition is not just intermediate between the healthy and the moderate. Actually, it has some unique features, which I think really adds something to the field, because there have been a lot of studies using single cell sequencing on the COVID-19 patient, but what they really focus on is they focus on the severe patient and try to understand what happens in the severe patient. But what we found that is that if you look at the asymptomatic patient, you can also find something very interesting. But of course, this study has quite a lot of limitations as well, because as you can see that we have a very small cohort size. We only have one severe patient, although severe patient is not the focus of this study, but still we only have 2 asymptomatic patients, and it's not really a gender and the age balance. And we also have limited temporal sampling for these 2 conditions as well. And so as a result, we also plan to add more sequencing to sequence more samples, especially from the asymptomatic patient. And at last, I want to thank my collaborator, Luyi Tian, for giving me the opportunity to collaborate on this wonderful project. And we also want to thank patient to kindly provide samples. And this is a bioarchive link. And welcome if you have any feedback or comments, feel free to contact me. Thank you.
Yeah, thanks for the talk. We have 2 minutes for questions. So from the audience, if you have any question, please just type in the Q&A chat panel of the Zoom meeting. So Luyi, I have questions about the 2 patients, the asymptomatic patients.
Yeah.
Are they all more or less the same, or these have some differences in, say, in the age or in their health condition or something? Or
for the clinical aspect, there's not much differences. They're both under 20 years old. They're both female, and they're quite healthy. From what I know, I didn't see they have any other clinical health issues. And also their result from the result that I showed, that also some of the result from these 2 patients looks quite similar. For example, the NK subsets also all very high for these 2 patients when you collect sample at different time point.
Okay, thanks. I don't see any. Yeah, there is one question. Why is there a preferential TCR expansion in moderate infections? Is it true for BCR also?
For the biological reasons, I won't say we're not sure. It's not surprising because in a moderate patient, when you compare to severe conditions, we expect patient to have a better immune response to the virus. So we do expect more clonal expansions, but what we are surprised with is we don't see much clonal expansions in asymptomatic patients. That's something we don't know. And also we mentioned that in our preprint, but due to the limit of time, we didn't show that here. But we do see clonal expansions in a B cell, especially in a severe patient. So the conclusion is the severe patient seems to have more clonal expansion compared with all the other conditions. And I think that finding is consistent. It's consistent with other recent published papers, as they also find in the severe patients there's a lot of clonal expansions in the BCR in the severe conditions. Yeah. Thank you.
Okay, thanks for the answer and thanks for the talk. Again,